Hello, and welcome to my video about GPCR mechanism for initiating the breakdown of glucose in the cell. First, we're going to talk about what exactly the GPCR mechanism is. So, cells have to have a way to communicate with each other, and being from trying to communicate to cells that are far away, to close, to right next to each other, they all have to have a different kind of mechanism. So the GPCR mechanism is a really common one used by cells. And GPCR are the G protein coupled reactions. They give off a signal with the help of G proteins. So this is important because it allows the cells to utilize signals that wouldn't be able to pass through the membrane. Usually this is because the signals are really large or they are hydrophilic, so they can't pass through the fatty part inside of the membrane. And then G proteins themselves are known as guanine nucleotide binding proteins. They're actually a formation of ATP. I will talk about those some more a little later on. So GPCRs, they're involved in the everyday basic functions. Like I said, they are one of the most common used mechanisms for cells to communicate with each other. So it's really common for them to be used for light-sensitive compounds, odors, such as like pheromones or hormones, or even neurotransmitters. So they're really great because they have such a wide diversity of receptors that can interact with different things and provide different functions to the body. So now I'm going to talk about a little bit of real-life connection just to put things into perspective. It is actually the case that GPCRs are used as targets for 40% of the medicinal drugs in the world, such as Abilify. So Abilify is an example. It's one of the most common bought drugs in the world for schizophrenia and bipolar disorders. So you can see just how important that these mechanisms are and how important it is for us to understand them, because they have allowed us to be able to target different kinds of diseases or cases utilizing or taking charge of these kind of receptors. So first, let's kind of talk about the structure of the GCPR. So it has two ends. You can see here, this one is exterior, so it's outside of the cell, and then we have one here inside the cell in the cytosol. So what's important here is that you'll actually see that it wraps in and out of the membrane, and it's going to do this 7 times. So we have 1, 2, 3, 4, 5, 6, 7. So in and out 7 times. And those tubes there are more just to emphasize that it's going through 7 times. It's not what the actual structure is, it's just for a visual. But just kind of give you an idea. So what's important here is this end here in the exterior is where the receptor is. So this is where molecules that wouldn't be able to pass through the membrane are able to interact. And the tail here is what's going to cause the next change, whether it's a second messenger or changing something next to it or that sort of thing. So to do a checkpoint, how many times does the GCPR receptors go through the membrane? And if you didn't go back, the answer would be 7. So now we're going to talk about the G protein structure. It has 3 main subunits, and then it will either be bound to GDP or GTP. So when it's bound to GDP, that makes it inactive, so it's not doing anything. And then when it's bound to GTP, it becomes active. So these 3 subunits, we have the alpha subunit, which is one of the most important because it is what's going to bind to GDP or GTP. We have the beta subunit, and then we have the gamma subunit. And then here we have GDP attached to the alpha subunit. Then this entire structure would be considered inactive. So before we go on, because we're about to go through the whole mechanism, let's just kind of talk about what glucose is, because that is what we're talking about, how the GPCRs are used to initiate the breakdown of glucose. So it's a sugar, and it's a primary source of energy in the body. It's formed during photosynthesis by water and carbon dioxide with some energy from the sun, so obviously that's occurring in plants, but we do consume it. It's incredibly important during cellular respiration. It's stored as starch in plants and glycogen in animals, so the process that we're about to see, you'll see that it refers to glycogen. and that's what initiates the breakdown of glucose. It's used during glycolysis to do anaerobic, aerobic, and fermentation. So that's really important because you can see how valuable it is to the cell and that it's used in almost every single kind of process to produce energy in the cell. So step one of this, so we have our GCPR chilling, and it has its receptor, so some kind of signaling molecule is going to come in, so whether that's again, it could be like a neurotransmitter, a hormone, that sort of thing. It's going to come in and it's going to attach here to the receptor portion that's in the exterior point of the GCPR. Next, we're going to see that it's going to release a signal here at the end that's going to cause GDP to be replaced on the alpha subunit by GTP. So it comes in, and it's going to kick it off and replace it. So, a quick checkpoint again. What is the active site of a G protein, and what is considered the inactive stage? So, inactive is when the alpha subunit is bound to GDP, and active is when it's bound to GTP. So next what we're going to see is the alpha subunit and GTP are actually going to separate here from the beta and gamma subunit. And then what's going to happen is they're going to interact with adenyl cyclase, right there, that just emphasized. And that's going to do is it's actually going to initiate an increase in production of CAMP, or CAMP. So we see that here, the adenyl cyclase is going to pop out a bunch of popping out a bunch of these CAMPs. So CAMP is really important because that's going to be the initiator of the next step. So CAMP is considered a second messenger. So here you see we have CAMP. It's going to initiate pKa, which will cause, the pKa has two subunits, with the catalytic units, the ones that allow it to phosphorylate other items around it. So that's what it's going to do. The CAMP is going to release those subunits of the pKa. This is then going to initiate our work with glycogen. It's actually going to activate glycogen phosphorylase, which then in turn will cause the cell to initiate the breakdown of glucose, which equals energy, which is the process that we were looking for. Now we're going to do some multiple choice questions just to review the topics that we've covered. So the first one that I have is adenyl cyclase releases what? Which our options are pKa, glycogen, CAMP, and GTP. So let you think about that for a moment. And the correct answer is CAMP. So remember that it's initiating the G protein will initiate the adenyl cyclase next by, which increases the production of CAMP and overall increases the breakdown of glycogen for energy. So the next one I have for you is, what are the three subunits of a G protein? So you have alpha, phi, gamma, alpha, beta, gamma, phi, sigma, alpha, beta. Now, the correct answer was alpha, beta, gamma. So remember that the alpha will break away from the beta and gamma subunits when its GDP is removed and a GTP comes into place to make it active. Next one I have is CAMP acts as a second messenger to what? So your options are pKa, glycogen, GTP, and GDP. The correct answer is pKa. So remember, an increased CAMP production, then it's going to initiate and take off the active part of pKa to then increase the breakdown of glycogen. The last one I have is that glucose is relevant in what kinds of production of energy. So you have anaerobic, aerobic, fermentation, and all of the above. And the correct answer is all of the above. So remember, glucose is super important. It's involved in so many different parts of the body and the cell energy and everything that's really just going on. So that's the end of my video. Thank you for watching. If you have any questions, please feel free to comment below.